studying for all my classes.
You are?
I really enjoy it. It's my last one.
Less and less people. Okay, guys. So who would still come to class even if they knew they were getting an A and didn't have to take the final? Okay, thank you. Now you're all raising your hands because you know I'm judging you, but I appreciate catering to me. Okay, so let's start out with extra credit. So you sent this paper. This was really nice. It shows all the different mechanisms for determining the cleavage plane. That's basically what the question was the other day, is what determines the cleavage plane to make sure that daughter cells separate during cytokinesis, the daughter cells are equal size. And there's a lot, like they go through yeast and bacteria, and of course, you know, I'm going to care most about eukaryotic cells. But there are a lot of mechanisms. Anything from, it has to do with the location of the astromicrotubules and the position of the mid-spindle, which is logical actually, to like in budding yeast, there are scars from where previous divisions have occurred, and it has to do with that, and so it's based on locations of prior divisions, and in some bacteria it was based on each division was perpendicular to the one before, and that all has to do with these landmark proteins that bind in certain areas and help to position the future contractile ring. And then some people told me about activation of Rho proteins, and that's true for at least for eukaryotic cells. And a lot of people talked about actinomycin. So if you hit any of these things, you're going to get a point. I'm a little behind on extra credit points. Don't worry, I'll catch up. It's this one and the prior one that still need to get up on Blackboard. What else? Yeah, so it was a good question, and there was a lot of reading involved, but hopefully everyone learned a little bit something. It was pretty obvious it was going to have something to do with activation of the Rho family of proteins, right? Pretty obvious, and it really just had to do with what is bringing the Rho proteins to where they need to be, so they can activate formins where they need to be activated, so that the actin can be polymerized in the correct location. Okay, is everyone happy about that? Most people got it. Some people are still just copying and pasting things and sending it to me. I don't give extra credit if all you do is copy and paste something. You get the extra credit when you actually type something out yourself. Even if you're sending me the reference. Like if you had an assignment from a professor and you gave them a published paper, would they give you credit for that? No, right? So the good thing is people are getting very good at answering the question. In the beginning of the semester, people weren't very good at that, and now you guys are pretty much spot on. I didn't really get at many people who are saying strange things that had nothing to do with the question, so that's good. Okay, so far we've been talking about cell cycle. We started talking about the different phases and the checkpoints. And then we started talking about regulation by cyclin CDK complexes and the regulation of those cyclin CDK complexes in terms of different phosphorylations and CKIs. And we spoke about the regulation of S phase in terms of presence or absence of inhibitors, presence or absence of activation of APC or activation of SCDK. Then we spoke about regulation of M phase in terms of mitotic spindle assembly, and what happens if things aren't going well. And then we spoke about entry into G1 phase and how you need low levels of cyclins, and how the activation of different forms of APC helps to make sure that APC stays on and M cyclin levels go down enough so that G1, the cell can go into G1. And then we started talking a little bit more about cell growth, because that's what this is all about, right? In a way, right? So the whole point of the cell cycle is so you can have more cells, right? So you can grow or to replenish. And so I brought up this question at the end of last class, right? A fertilized mouse egg and a fertilized human egg are the same size. What factors are responsible for the huge difference in size of the adult mouse and human? And we talked a little bit about this and came up with these three things, right? Cell growth, cell death, and cell division. Those are what determine the size of the organism in the end, right? If you have cell growth, high levels of cell growth, not too much cell death, lots of cell division, you're going to have more cells, right? So, and this is my segue into connecting cell cycle with signaling. So we're going back to signaling. Does everyone remember signaling? Yeah, you thought you were done. See, I keep doing it to you. It's fun. So you have to think about cell cycle not as just something that happens automatically, but it's something that's regulated. It's something that needs to be controlled, right? So it's not just like there's this clock that's going, okay, time for G1, okay, time for S phase, right? This is all, this can be activated by something called mitogens. Mitogens are signaling proteins that are outside of the cell, and they bind to receptors and stimulate cell division. So they actually stimulate entry into S phase. And this is how they work. So here's your cell surface and your receptor, and your mitogen will bind to your receptor, activate RAS. Does everyone remember RAS? Broken nail. It's annoying. Remember RAS? What does RAS do? Amplifies signals, right? So what kind of protein is RAS? GTP binding protein, molecular switch, helps to turn on signaling, remember? Amplifies, spreads out signals through different pathways, right? So when a mitogen binds to a receptor, it activates RAS, which then activates MAP kinase. Right, and so then some gene regulatory protein goes into the nucleus, and it activates gene expression, resulting in upregulation of a whole bunch of genes, one of which is MYC. Who's heard of MYC before? Most people have. Like, if you had to ask a freshman what genes they've heard of, most of them have heard of MYC and P53. There's some big ones, usually because of their involvement in cancer. So then you get activation of MYC, which MYC is a transcription factor which activates gene expression. So you have more genes being turned on, which will result in increasing G1 cyclins. So you get G1 CDK activity. Okay, so now you have an active CDK, G1 CDK, and what do CDKs do? What's their job? They phosphorylate things, so you have to say what's it phosphorylating. It's going to phosphorylate RB. So right now, RB is an inhibitor, and RB binds to E2F, which is another transcription factor involved in activating gene expression of genes required for entry into S phase. So right now, RB is holding on to E2F, keeping it inactive, right? Because you don't want to go into S phase if it's not time. And G1 CDK comes along, phosphorylates RB, changes its conformation, changes its shape, and then it goes away, and liberates E2F. And now E2F can go on and induce gene expression of a whole bunch of genes, many of which activate S phase. Okay, so we'll go through this a little bit more, right? So you got mitogen, it binds to the receptor, activates RAS, MAP kinase, magic happens, remember the signaling magic, relaying signal to the nucleus, some gene regulatory protein goes into the nucleus, binds to some promoter, actually a whole bunch of promoters, and activates a whole bunch of genes, one of which is MYC, right? So one of them is MYC, which then binds to other gene, it's a gene regulatory protein, which means transcription factor. It binds to other promoters, activates expression of other genes, and one of which is the G1 cyclin, which binds to the G1 CDK and it activates it. And then the G1 CDK phosphorylates RB, so it changes its shape, looks like a mustache, right? Do you think RB looks like a mustache? Those are the eyes. Kind of looks like a mustache, no? I feel like there's been a lot excessive use of mustaches in protein shapes in this book. What do you guys think? I think that at least twice I've seen proteins drawn looking like mustaches. Anyways, or I'm having a flashback to last time I taught this class. So that goes away, and it allows E2F to be released and have activity. It activates S phase gene expression, which then activates other cyclins, including induces expression of S cyclins or G1S cyclins, which activate SCDKs. What happens when an SCDK is active? You enter S phase and you get replication of your DNA, right? And so that is how an extracellular signaling protein will activate, will push a cell into cell cycle. Does everyone like that? So RB, just for people to be interested in RB, RB is the retinoblastoma gene, and it was originally identified in retinoblastoma, which is a kind of cancer, where people who had completely lost RB, so both copies of their RB gene were deleted or mutated or something were not active, those people developed this tumor. Logical, right? Because what does RB do? It inhibits going into S phase. So if you can't inhibit going into S phase, what are you going to do? Go into S phase, right? If you go into S phase, then you're going to go into cell cycle and you're going to divide eventually, and then you're going to become cancer if that keeps happening in an uncontrolled way. Is everyone happy with that? That's nice and neat. Don't you love that? Okay, good. Okay, so based on what I just told you, let's do a little review. How do mitogens promote cell cycle entry? I just told you. Just spit it back at me.
Because it activates RAS, which sends other signals. It activates MAP kinase, and then sends signals to the nucleus, and then it upregulates gene expression, specifically of MYC, and then
Yep. So what does MYC do?
it increases G1, the expression of G1 cyclins.
Right, so MYC activates gene expression, which results in activation of G1 CDKs, which what does the G1 CDK do? Phosphorylate RB, which when RB is phosphorylated, it does what? E2F, right. It releases E2Fs, which can go on and basically activate more gene expression, allowing SCDKs to be active and starting S phase. So you don't need to memorize that whole thing. You could just say it's a signaling pathway that activates MYC, which then activates S cyclins. Now, well, actually, you want to keep RB in there. You want to talk about RB. So What role does RB play in cell cycle control? What role does RB play in cell cycle control? Yes? It stops from going into S phase. What does it do to stop going into S phase? It inhibits E2F. How does it inhibit E2F? How does RB inhibit E2F? Really easy. Yep. It binds to it, right? If you have a protein, remember proteins have shapes. They have these domains that do things. So if you have another protein that comes and binds to it and prevents it from interacting with whatever partners it needs to act with, then it's gonna inhibit it, right? Okay. So what does E2F do to promote cell cycle entry? What kind of protein is E2F? I hear whispers. A transcription factor. Transcription factors regulate gene expression, right? So what kind of genes does it regulate? S cyclins and other genes required for S phase, right? So it's going to bind to a promoter. I know not everyone has taken 409, and I don't talk about gene regulatory proteins, but you can just know and understand the fact that gene regulatory proteins, aka transcription factors, tend to bind to the region upstream from the transcription start site and help to recruit RNA polymerase. Okay, so you can imagine how those genes could be activating or inactivating, but in this case, E2F promotes cell cycle entry, so it binds to all these promoters, these regions just upstream of the genes of interest, and recruits RNA polymerase so that it can start transcription. And that's how it works. Okay, so you're studying a new protein, which you believe is involved in S phase entry. You mutate it and analyze the effect on cell cycle entry. You find that in cells containing the wild type protein, 50% of cells are BRDU positive compared to cells containing the mutant protein, where 100% of cells are BRDU positive. What is the likely function of the protein, and what effect is the mutation having? So first you have to think to yourself, what's BRDU? What does that do? What does that mean? So why don't you guys talk to each other for a minute about this, and we'll reconvene and go over it. Uh-oh, no one's talking. Is everyone tired of talking to their neighbor? Do we want to change seats so you can have new neighbors? Yeah, some people like their neighbors. Okay. There you go, for those who are visual learners. Okay, since it's so quiet, it could be quiet just because there's no one coming to class anymore, but I think it's quiet because people want to know the answer. So what does BRDU do? Yeah.
I'm not exactly sure what it does, but I remember on the quiz question, it had something to do with like recently passing through S phase.
It has something to do with recently passing through S phase. Okay, so you are right, but what does it do? What is BRDU? It stands for bromo something uridine. Yep.
It integrates, like, it's like a marker that integrates into the new DNA.
Right. So it's something that integrates into the new DNA, also known as a nucleotide analog, right? So it's not a normal nucleotide that's being used when doing DNA replication, but it looks enough alike that it fools the cell so that it gets incorporated where the T's should be. So it's a thymidine, right? Analog. And, right, so you can see how it would be incorporated during S phase, because during S phase, the cell is taking its nucleotides and putting it into the new DNA, right? Yep. So, and then you can use an antibody, right? So you've got your cell with your nucleus and your DNA, your one DNA, and then everywhere the T should be, B's go in, and then you do, after you are finished with your experiment, you take an antibody, and the antibody recognizes that, and it's red or whatever fluorescent color you want it to be, right? And so all the cells that have integrated their BRDU into their DNA are going to be red, and those that haven't won't be red. So cells that are BRDU positive are what? What did they just do? They just went through S phase, right? Okay, so now go back to the question, which is, what is the likely function of this protein, right? So wild type protein, 50% were BRDU positive, and this protein that you mutated somewhere special, 100% are BRDU positive. So what do you think was mutated and where? Or what do you think is going on here? Does anyone have a hypothesis? Yeah. Okay, so S phase is happening when it's not supposed to, probably for some of the cell. Because your wild type is your control. So you know from your control that at any given time, about 50% of your cells have gone through S phase. And with your mutant, they've all gone through S phase. So some kind of control factor, some kind of regulatory factor was taken away, or the function was taken away. So what do you think? What do you think this protein that you mutated? What do you think it was doing? Yes. It was probably an inhibitor, right? Because if you take away an inhibitor, then it will promote something, right? So that could be your 100%. What kind of inhibitor did we just talk about? RB. So does that fit the mold? If your RB is not working, then E2F is free to push cells into S phase, right? So what kind of mutation do you think you made that would result in RB not working? So now you have to think, okay, how does RB work? What does it do? What regulates its activity?
Phosphorylation site. This is mutated.
So maybe you mutated its phosphorylation site, right? Now, ideally, if you're a scientist, you know what mutation you made. You're not just randomly making mutations. But you can easily make this like a patient. You're a doctor, you're an oncologist, and you're studying patients that seem to be developing a certain kind of cancer, and you know it has to do with RB. Chances are they've accumulated mutations that have affected its ability to be phosphorylated. And that's usually what you see in a lot of cancers, where signaling proteins or inhibitors, or as we'll see in a moment, P53 is mutated. It's usually sites that regulate the activity, which are often phosphorylation sites. Okay, good. We got through it, guys. Good job. Okay. DNA damage prevents entry to S phase and mitosis, right? Does that make sense? You don't want to replicate damaged DNAs and DNA, and you don't want to segregate damaged DNA to daughter cells, right? So if you were a cell and you were trying to protect yourself or your organism, not like a cell is protecting its organism, what would you do if you had DNA damage and you don't want to go into S phase, you don't want to go into apoptosis, you don't want to go into mitosis? What would you do? You could do two things. Apoptosis was one answer. What was the other? Remember, we have all these checkpoints and stuff like that. Okay, so you'll activate the DNA damage response, which those who had 409 are aware of. Actually, you guys learned in 309, so you remember the DNA damage response. But what does the DNA damage response do? Cell cycle arrest, right? So it stops the cell cycle, allows the cell to repair. What else does it do? What if the damage cannot be repaired? Apoptosis, right? So cell cycle arrest and apoptosis. Okay, so this is how it happens. So yeah, you're absolutely right. If you have DNA damage, you're gonna activate the DNA damage response, which I have not taught you about, but I will next semester for those who want to stay with me. And that results in, so here's your X-ray, your DNA damage, and then you get these foci that occur at the points of damage, and they activate kinases, ATM, ATR, check 1, check 2. Doesn't matter for you guys. It activates kinases. And by now you should know it's all the same, right? You activate kinases, they phosphorylate something that affects something, right? That's pretty much all of cell biology. So here you activate these kinases that phosphorylate P53. You guys remember hearing about P53. P53 is one of the most mutated genes in cancers. Find me a cancer with wild type P53. I bet there's some, but I bet there's a lot of them that have certain mutations. Anyways, when you phosphorylate P53, you change the shape, and that prevents MDM2 from interacting. MDM2 is a ubiquitin ligase. MDM2 is constantly ubiquitinating and degrading P53, so P53 levels in the cell are nearly undetectable. If you take any cells and you do a Western blot for P53, you should not be able to detect it, which is actually what makes sometimes studying P53 difficult if you don't realize that, because you need to. So I once worked on P53 for a short period of time, and we were using these cells, and we were like, we can never see P53. Well, duh. that's the cell working how it should. So when you're doing research, you always need to realize what you're doing before you do it, because sometimes you do really dumb things. So we had to get these special cells that overexpress P53 just so we could study what was going on. Anyways, MDM2 constantly ubiquitinating and degrading P53 in the proteasome. You get phosphorylation of P53 when there is DNA damage, which changes the shape of P53 and doesn't allow it to interact with MDM2 anymore. And P53 is a transcription factor, which means it binds to regulatory regions of genes, such as promoters, and activates gene expression by recruiting RNA polymerase. So you get transcription, translation, and one of the genes, so P53 activates a whole bunch of genes, but one of the genes in this example that's activated is P21, which is a CKI. So it's kind of logical, right? DNA damage, you want to stop cell cycle, right? And so one thing that you do is you'll activate expression of a CKI that inhibits G1S CDK and SCDK. So if you inhibit your CDKs that push you into S phase, then you're not going to go into S phase, cell cycle arrest, right? Makes sense, right? Okay, so let's just go through this a little bit more closely, right? So normally P53 is kept at really low levels by MDM2, ubiquitin ligase, and when you phosphorylate P53 by these DNA damage activated kinases, CHEK1 and CHEK2, then it prevents MDM2 binding, and if the ubiquitin ligase can't bind to its substrate, then it can't degrade its substrate, so now it's going to be stable. And so P53 is stable and it's active, and it can go on to activate gene expression. And it activates P21, CKI, and that will bind and inhibit G1S or STDKs, so they can't push the cell into cell cycle. Any questions? I know, it's so easy, right? Okay, so you're studying P53 mutations, and you find a mutant form of P53 that has a slightly lower molecular weight than wild type P53. So what's the molecular weight of P53? Thank you. 53. Did you guys know that when proteins are named P something, it's protein and its molecular weight? So P100 is 100 kg, P53 is 53, P21 is 21. And another special treat is if you want to know how many amino acids are in a protein, just add a 0 to the molecular weight, and it's a decent estimate. So if you've got P53, you can assume it's going to be about 530 amino acids. That's a fun trick. P21 about 210. Anyways, okay. So you find that it has a mutant form of P53. I didn't learn that special amino acid trick until I was in grad school, and it's really useful. I'm just saying, for those of you who are going to go do research, if you're doing a Western blot and you have your antibody and you're like, oh, what size is this protein I'm looking for? And you lost the sheet that came with the antibody, and your computer's not working, and you have no way to look, your iPhone is in your car, you have no way to find out the size of the protein. Well, if you let me take that back. You're going to know because it's called P53. It's 53 kD. But there was another reason. It was helpful. Oh, right, in knowing how many nucleotides, because then you just multiply by 3, and that's helpful too if you're running a gel. Right? 53 times 3, because there's 3 nucleotides per codon. Yep. Anyways, I digress. You guys are enjoying the end of the semester, aren't you? Okay. So you find a mutant form of P53 that has a slightly lower molecular weight than wild type P53. Right? So wild type P53 is 53, or 53 in a little bit. And this one's like 53 and a little less. And cells containing this mutant P53 have twice as much PI staining. Oh no, she's saying this is another method, PI staining, as wild type cells after exposure to X-rays. What is a likely explanation for this observation? So you've exposed your cells to X-rays. This is going to do something to P53, right? Just based on what we've learned. Some cells will have a slightly higher, some cells will have a slightly lower molecular weight dependent upon this mutation. So what's going on here, and what's an explanation for this observation? So you can talk to yourselves, and I'll come up with some kind of thing to draw on the board that makes sense. Talk. At least pretend.
That's something that has more PI stating than just guessing the entire concentration of the cell.
I know I don't bring that up. It's just
You sound negative. You can sound negative. I know if you. That's for the
Okay, see if this works. Okay, so here's the gel you ran, right? So you've got your marker, which shows you your sizes. You've got your control, so these are cells not treated with X-rays, and they're cells that overexpress p53, so you can detect them. You did a Western with an anti-p53 antibody, and you saw in your wild type of mutant these proteins. Then you treated with X-rays, and you found your wild type is at this level, and your mutant is at this level. Okay, so then you're like, oh, interesting. And so then you did flow cytometry with PI staining, right? So this is cell number, and this is PI staining for both of them. And so your wild type, you see this, so this is your 1 and this is your 2, this is your 1, this is your 2. So for this one, you see most of them are stuck in what phase? G1. And in this one, a lot of them are where? S. Well, this is S. So G2, so they've completed S. They've done G2 and have not finished M yet. Okay, so that's your data. What happened here? What's a likely explanation for this? What's your mutant? Where did you mutate? Where's your p53 mutated? Okay, so let's go back to this. What regulates p53 activity? MDM2. So what prevents p53 interaction with MDM2? Phosphorylation, right. So when you have DNA damage, like X-rays, you're going to end up with phosphorylation of p53, and that's going to result in what? Sometimes apoptosis, but in what I just told you guys, gene expression of what kind of gene? Genes that prevent entry into cell cycle, right? Because you don't want to do that when you have DNA damage, right? Like CKIs, okay? So here you've got this p53 mutant that you know nothing about except for the fact that when you treat your cells with X-rays, your wild type does what it's supposed to do and increases in size. Why is it increasing in size? What happens when you activate p53? It's phosphorylated. Do you think phosphate groups change the size of a protein? Right? So if you add phosphate groups to a protein by phosphorylating that protein, it's going to migrate more slowly. So that's your phospho p53. Your mutant, what's happening here? So would you say it's not being phosphorylated? Okay. So right now you're like, oh, it's not being phosphorylated. So the next thing would be, well, can it activate gene expression? Can it stop the cell cycle, right? And so that's where you look at this, right? Your wild type, when you treat with X-rays, your p53 should become activated, which you saw here it does, and it should activate expression of CKIs, which should prevent entry into cell cycle. So you should be stuck in G1, which you are. In your mutant, though, that's not happening, right? So you're not able to inhibit entry into cell cycle. You're not able to activate P53. You're not able to activate gene expression. You can't make CKIs, and so you don't stop cell cycle, and you keep going. And this is what cells normally look like. You have some cells in G1. Actually, you have even more here. Some cells in S and some cells in G2M. You guys follow a little bit? These are the kind of questions I like to put on exams. So it's definitely helpful if you either pay attention and understand it or ask questions if you don't. Right? Okay, so everyone loves it. Yes? What happened to the mutant? Okay, so you see how the wild type is higher? So it's phosphorylated. And the mutant's not higher, right? Because X-ray, so let's go back to this picture. Right? So when you treat with X-rays, what happens, right? Phosphorylation, you activate all these kinases, which ends up in phosphorylation of P53, right? So X-rays, phosphorylation of P53 because it's higher. Mutant, no phosphorylation. So there must be a mutation that's preventing phosphorylation. Possibly a mutation in what site of P53? What site of P53, what amino acids of P53 do you think ended up being mutated? So possibly ones that are phosphorylated. You guys are killing me today. Am I completely speaking Chinese? Who knows Chinese here? Anyone? No? Who watches Ni Hao Kai Lan? Does anyone watch Ni Hao Kai Lan? Okay. So you know a little bit of Chinese. Okay. When you have kids, you'll know. Okay. Anyway. So I'm speaking Chinese today. So phospho P53 cannot be phosphorylated. Remember, we're talking about a mutant P53 that we're studying, right? First thing you're going to do is see, well, can it be phosphorylated? It clearly cannot. If you wanted to follow up on this, you'd run another Western and use the phospho P53 antibody that can only detect phospho P53, and you would see that you get one band, and that's it on the whole gel. And so then you do PI staining to see what part of the cell cycle the cells are in response to treatment with X-rays, right? Because if you know X-rays are supposed to result in what? Gene expression. Yeah, DNA damage response. Expression of CKIs. CKIs prevent activity of whatever cyclin CDK complex. In this case, it's the G1S and S CDKs. So it's logical that that would be something that you can see a difference in if your P53 is not being activated, okay? And so normally, if you treat with X-rays, you should not, you have a lot of CKIs, they're going to prevent your SCDKs. You're going to be stuck in G1, right? This is, remember, G1, this should be S, and this should be G2M, right? G1, S, G2M. So the difference is the P53 that cannot be phosphorylated, it's that basically the DNA damage response is not being activated at all, right? You're not expressing any CKIs because you're going into S phase and G2M just fine. Okay, see how there's no block. There's no cell cycle arrest. Questions? Everyone's clear? Okay, let's move on. Okay, so when DNA damage is too severe to be fixed, you get cell cycle arrest or apoptosis. And this will be, we're going to start talking about apoptosis on Friday. But, so remember, you've seen this before, right, where you have PI staining, and that's staining your DNA, right? It's your DNA content, and you'll get this G0, G1 here, they have it here, S phase, and your G2M. So that's how you're content of the DNA content of your cells vary based on what's going on with the cells. And then if you've got these cells all the way down at the bottom with almost no DNA, those are your apoptotic cells. So we'll talk about apoptosis shortly. So one last thing before you go. Excessive cell proliferation can signal another pathway, which results also, remember the same thing where you have MYC activation. Remember I said MYC induces a whole lot of things. And so one thing it will do is it will activate ARF, another gene. Remember, MYC is a transcription factor, activates expression of many genes, activates ARF. And what ARF does is it does the same thing from the other side. It binds to and inactivates MDM2. Remember, MDM2 is the ubiquitin ligase that sends P53 to be degraded. And if you take away the ubiquitin ligase, so rather than phosphorylating P53 and preventing interaction, you're actually then competitively binding to the ubiquitin ligase, preventing interaction. Both stabilize P53, right? And this results in cell cycle arrest, and it could also induce apoptosis. Do you guys want to see my apoptosis movie before you go? Who said no? Who said no? Come here so I can hit you. Who said that? I love it. Everyone feels really comfortable with me. That's good. That's good. Well, let's just do it for fun. There's music involved, but you're gonna miss it.
Apoptosis, a form of programmed cell death,
Ooh.
has been induced in these cultured cells. Cell death is characterized by blebbing of the plasma membrane.
Yeah, you're not gonna hear it. It's
fragmentation
okay.
of the nuclei.
You don't need to hear it. Let's see what-
Suddenly, cells weaken attachment to the
Let's hear about
substratum
that.
that they have been growing on, shrivel up without lysing.
These are nice, happy cells. Apoptotic cells shrivel up and.
In the following movies,
How
we
they
observe
die.
the process at higher magnification.
I know, it's really nice. I know, I hope everyone can hear the music. That's a nice high magnification. And you see there's this blebbing that's characteristic of apoptotic cells. See that blebbing? And what the cell's doing is it's keeping its DNA contents enclosed in membrane because it doesn't want to activate an immune response. Doesn't want to let things out. Often viral infection can induce apoptosis too as part of the immune response. You don't want to let the virus out. So you encase everything in membrane. Isn't that fun?
Yeah, exactly. That's what I was just thinking.
Okay. You guys can go away.